Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this.
Hey everyone, welcome if you're new. Welcome back to my awesome repeat listeners. Today is the highly awaited, much anticipated calcium episode that I only hope can live up to all the hype I've given it. Calcium is so important and it's such a cool signaling molecule that I am super excited to get to talk to y'all about it. So without further ado, let's talk about calcium. So where is the calcium in your body? It's mostly tied up in your bones and your teeth. There's only about 3 millimolar in the serum and half is bound to proteins, so your free calcium is about 1.5 millimolar, which is low, but it's astoundingly low inside the cell. It's only 50 nanomolar, so this is orders of magnitude different, which is much bigger difference than in any other ions that you have in your body. And so the reason intracellular calcium is so low is because calcium and phosphate, if left to their own devices, will actually precipitate out of solution and essentially would turn the cell into a pebble. Now, at some point along the evolutional tree, evolution decided that intracellular phosphate was more important because it's hugely important for ATP, for DNA, all this kind of stuff. So the cell chose to exclude calcium. And if there's any significant amount of calcium in the cell, you will actually get precipitation, which is wild. But this low calcium concentration inside the cell is actually now extremely useful because it's the ultimate signaling system. Any calcium that comes in is going to make a big difference because the cell is so hypersensitive to it. It's always going to notice it. Calcium also binds proteins really well, so there's lots of calcium sensors inside the cell. And being a cation means that flowing in causes a depolarization, and then once it's inside, it's also a chemical signal. So it converts an electrical signal to a chemical one. It's a very cool signaling molecule. So in general, the workflow of calcium is that there's some kind of trigger that releases it, then it binds to a sensor, and then there has to be an off switch to put the genie back in the bottle because, like we said, calcium floating around the cell is very dangerous. Now, the release of calcium can either come from the outside or it can come from internal calcium stores. From the outside, it usually comes in through voltage-gated calcium channels. Sometimes there are ligand-gated channels like in the brain. There are also store-operated channels, which we'll talk about, and then there are leak channels, which just means channels that we don't really know how they work yet, so we call them leak channels. And then internally, the endoplasmic reticulum or sarcoplasmic reticulum, if you're in a muscle cell, are major calcium stores. The mitochondria can also store calcium, and endosomes may also have some calcium, and it mostly gets out by IP3 gated channels and ryanodine channels. Calcium, when it binds, likes to bind 7 oxygens, so it binds really well to carboxyl groups on glutamate and aspartate specifically. And we're going to talk about some specific motifs. So first of all, there's the EF hand motif. So this is a helix-loop-helix motif that's often found on calcium binding proteins like calmodulin and troponin. Calcium binds really nicely to it. And then we also have to discuss C2 domains, which is a protein domain that's made of 2 beta sheets. And they can bind acidic membranes with calcium. So basically what they can do is they recruit things to a membrane. Now the off switch for calcium, for fast signaling, calcium diffuses really quickly away from the mouth of a channel because the intracellular concentration is so low. So that kind of locally turns off the effect of calcium right where it flowed in. But then long term, there are several strategies for getting rid of it. So there can be extrusion, meaning pumping it to the outside via exchangers and pumps. So you can have calcium ATPases, which harness ATP directly. Those are basically pumps. And then there's also the sodium-calcium exchanger, which moves calcium using this energy from the sodium gradient. So that's an antiporter. Sodium flows in, down its concentration gradient, calcium gets pumped out. It can also be sequestered in the endoplasmic reticulum, and in that case, you're going to pay with ATP to use the circuit transporter because, again, the concentration inside the endoplasmic reticulum is much higher than the concentration in the cytoplasm, so you have to pump it. So you are using ATP in that case. And there's also a recently discovered uniport, which can allow calcium into the mitochondria for storage, which is interesting. But either way, you're going to be pumping across a fairly steep gradient, and it's going to cost you energy. And the cell also kind of short-term does buffering by having proteins bind to calcium. So 95 to 99% of intracellular calcium is going to be bound at all times. There's really not a lot of free calcium floating around for the reasons we discussed. So calcium does so many things, and we can't possibly discuss all of them. And as we go along, every time calcium comes up, you'll hear me get excited because I love calcium. But here we're going to talk about just a few of the really big jobs that calcium does. So first of all, synaptic transmission. If you recall, we mentioned maybe 2 episodes ago that calcium is in charge of getting neurotransmitters out of the presynaptic membrane so they can do their thing. And we said we would talk a little bit more about how it works. And we will talk a little bit more about how it works, but we actually don't fully understand the process yet, so I'll tell you what I know. So depolarization from an action potential is going to open voltage-gated calcium channels. These voltage-gated calcium channels are right next to the fusion sites of the vesicle. So the calcium is going to bind a protein called synaptotagmin, which is a protein on the vesicle membrane, and it has 2 C2 domains, which bind calcium near a membrane, remember, and they recruit something to a membrane. So by binding calcium, this causes the vesicle to be recruited to the membrane, and then the vesicle can kind of penetrate into the plasma membrane and fuse and allow the neurotransmitter into the synaptic cleft. We don't totally understand this fusion process yet, but we know it works. And we see calcium triggering secretion in other contexts also. The same kind of process happens with insulin-containing vesicles in the beta cells of the pancreas. We see the same kind of process with histamine secretion from mast cells. So we know that calcium likes to do this. And the off switch here is actually super cool because calcium is super cool, but there are calcium-gated potassium and chloride channels, which drive potassium efflux and chloride influx, both of which hyperpolarize the membrane to shut down the calcium channel. So this is basically a built-in negative feedback loop. The depolarization allows the calcium in, and as soon as it comes in, it starts binding to these channels and opening them up to repolarize the membrane and stop any more calcium from being allowed in. And then the cell can take care of the calcium in the normal way, pumps and buffers. Now, how does calcium cause muscle contraction? This works very differently in smooth muscle and in skeletal muscle. So let's start with skeletal muscle contraction. The terminology used in muscle cells is different than in other cells. I don't know why, probably just because they like to confuse us, but the T-tubules are the equivalent of the plasma membrane, and the sarcoplasmic reticulum is the equivalent of the endoplasmic reticulum. So just bear that in mind. Now, on the skeletal muscle, there are 2 calcium channels at the junction of the T-tubule and the sarcoplasmic reticulum. One is on the T-tubule and one is on the sarcoplasmic reticulum. The channel that's on the sarcoplasmic reticulum has a ryanodine receptor, which is physically associated with the voltage-gated calcium channel on the T-tubule. And this is important. So depolarization occurs when acetylcholine opens its channels and causes an end plate potential, which then triggers an action potential and depolarizes the membrane of the muscle cell. And this causes voltage-gated calcium channels on the T-tubule to open. Now, oddly enough, this is actually kind of a fake channel because it doesn't let any calcium in. Its function is to pop the ryanodine receptors open through its physical connection, and that's where the calcium comes from. It does not actually come from the outside. All the calcium comes from the stores in the sarcoplasmic reticulum. And then the calcium is rapidly pumped back into the sarcoplasmic reticulum by the sarcopumps. It doesn't use plasma membrane pumps very much to get out of the cell because that's not where it came from. Obviously, some is going to go out through that way, but most of it gets pumped back into the sarcoplasmic reticulum. And this release of calcium into the cytoplasm is what causes muscle contraction. How does it do this? Calcium binds to troponin, which is an EF hand protein. Now we know that muscle contraction is caused by interaction of actin and myosin. At rest, myosin binding sites are covered by tropomyosin. Now when calcium interacts with troponin C, it causes the tropomyosin to move, and the binding sites become accessible for the myosin to bind and do cross-bridge cycling. This is a tangent because we're not really talking about cross-bridge cycling, but we have to talk about cross-bridge cycling. That's when the myosin head binds to the actin, and it's also bound to ADP. And it kind of like pulls itself forward. We call this the power stroke. And then the ADP leaves and ATP binds in its place, which opens a cleft, and the actin and myosin dissociate. And then the cleavage of ATP allows the myosin head to rotate and attach to the next actin on the filament. And now it's bound to ADP because remember we just cleaved the ATP, and it repeats the whole process. And that's how calcium causes muscle contraction in skeletal muscle. In cardiac muscle, the process is similar. The only difference is that the calcium channel is a real one. Calcium actually does come in from the outside, and the ryanodine receptor in this case is not physically gated, it's calcium gated. So once calcium starts coming in from the outside, it also pops open. This is a huge, huge positive feedback loop. And it has a specific name. We call it calcium-induced calcium release because it's calcium release that's induced by calcium. And in this case, plasma membrane pumps move the calcium back out of the cell. It's not really sequestered as much in the sarcoplasmic reticulum. And drugs that target these pumps that pump calcium out are going to affect cardiac muscle over skeletal muscle because the plasma membrane pumps are much less important in the skeletal muscle. So the classical example of trying to control calcium in order to control muscle contraction is digitalis, which is in very old medication actually for heart failure. And what it does is it blocks the sodium-potassium ATPase, which decreases the sodium gradient and impairs the calcium-sodium cotransporter. So more calcium is going to stay inside the cell, which helps the heart contract more strongly. This is sometimes used in congestive heart failure. It's pretty old medication, like I said. We don't use it so much anymore because it has a pretty narrow therapeutic window and there's a lot of toxicity associated with it, but this is the principle behind it anyway. Now to talk about calcium in smooth muscle. So this is actually done through GPCR signaling. GPCRs that are coupled to G sub Q. Remember that what happens with G sub Q is that ligand binding activates phospholipase C beta, which cleaves IP2 to diacylglycerol and IP3. And this opens IP3 gated channels in the sarcoplasmic reticulum, releasing calcium. In some cells, there are ryanodine receptors, which do calcium-induced calcium release also, but not in all of them. Now, the calcium getting out into the cytoplasm causes muscle contraction a little bit differently than it does in skeletal muscle. So in smooth muscle, instead of needing tropomyosin to be moved, in smooth muscle, myosin needs to be phosphorylated to cause muscle contraction. So calcium binds to calmodulin, which is another EF hand protein, and calcium and calmodulin bound together can now engage a myosin light chain kinase and release its autoinhibitory domain so that myosin light chain kinase is now active. What do kinases do? They phosphorylate things. Myosin light chain kinase phosphorylates myosin, and then it allows muscle contraction. So what is calmodulin? Because calmodulin is fairly important. It's like we said, an EF hand protein, and when it's bound to calcium, it can fold up and it can bind to calmodulin binding domains on many different proteins, which turns them into calcium-gated molecules or calcium-controlled molecules, even though those proteins are not directly binding calcium. And one major class that it works with is autoinhibition release of kinases. So just like it does with the myosin light chain kinase, there are a lot of different kinases that are reactive to calcium in this way. Calcium binds to calmodulin, calmodulin releases the autoinhibition of the kinase. Now, something we have to discuss is store-operated channels. So in a cell type without voltage-gated calcium channels, you're going to be emptying your endoplasmic reticulum calcium stores because calcium is still going to get extruded from the cell by pumps. Even if some comes back into the endoplasmic reticulum, not all of it does. Some of it gets pumped out, but there's no obvious way for it to come back in. So eventually, the endoplasmic reticulum is going to run out. And this is where store-operated calcium channels come in. And these are so cool. I love store-operated calcium channels, and I'm going to try to explain them to you, but I just think they're awesome. So how do they work? So there's a protein called STIM1. It's situated in the endoplasmic reticulum, and it has an EF hand, which, remember, binds calcium very well, facing into the lumen of the endoplasmic reticulum. And when the endoplasmic reticulum is full of calcium, there's plenty of calcium around, the EF hand is bound with calcium. Now, we step away from that for a second. There is an inactive calcium channel in the membrane called ORI1. And when calcium drops in the endoplasmic reticulum, the STIM1, which is now not bound to calcium like it wants to be because there's not enough calcium there, starts to aggregate and it migrates to a site in the endoplasmic reticulum which is in contact with the plasma membrane. It contacts ORI1 and it actually pops it open, which allows calcium into the cell, and then CIRCA quickly pumps the calcium into the endoplasmic reticulum. And so we call this store-operated calcium channel ORI1 because it's only open when the stores of calcium are low, which I think is so cool. Now, calcium is also important in immune function. T cells use calcium. T cell receptors, when they're activated, drive an IP3 signal, which releases calcium. And then the calmodulin calcium complex binds calcineurin, which is a phosphatase. So we had talked about the calcium calmodulin complex binding kinases, which phosphorylate things. They also can bind phosphatases. And in this case, they do. They bind calcineurin, which is a phosphatase, which dephosphorylates a transcription factor, which can then go to the nucleus and turn on cytokine production and a signaling pathway similar to smooth muscle activation, just using a phosphatase in this case instead of a kinase. In some cases of severe combined immunodeficiency, there's actually been found to be an ORI1 dysfunction, leading to not enough calcium in the ER, which doesn't allow cytokine production. And this knowledge can be used therapeutically also. So in allograft rejection, which is where we're worried about rejection of a transplant, cytokines cause a large part of the issue. So there are immunosuppressants that target calcineurin specifically, which ensures that NFAT, which is the transcription factor that it dephosphorylates, can't be dephosphorylated and therefore can't start cytokine production.
Okay, one more use for calcium. The endoplasmic reticulum and the mitochondria are connected, and these contact spots are really hot spots in the mitochondria because the endoplasmic reticulum can feed calcium to the mitochondria into the outer membrane, which is super porous, and then there's a uniporter called MCU, which allows the calcium into the inside of the mitochondria. And elevated calcium can drive dehydrogenases and oxidative respiration, but it's also a signal for apoptosis. We will talk about apoptosis a little bit later. We don't understand exactly how or why calcium can give these mixed messages and what causes the calcium message to be interpreted in different ways, but we know that calcium is super important in this process, which is not surprising because calcium is just super important. And now, last point I want to make about calcium signaling is that the time scale of calcium signaling can be very widely varied. Calcium can cause neurotransmitter release, for example, which is fast, or it can cause changes in transcription, which is a little bit slower. Calcium has so many different uses because it's such a super diverse molecule. Okay, this was a whirlwind tour of some of the awesome powers of one of my favorite mighty ions, and there is more to talk about because there's always more to talk about, but this will be a really useful foundation to rely on when you encounter calcium down the line. And don't worry, I will reference back to this plenty, so if you stick with me, I will not let you forget this stuff about calcium. Thank you for listening. Hopefully you learned something and you came to love calcium just a little bit today. If you enjoy the show, please rate it and share it with your friends. And always feel free to reach out to me by email, medtogether26@gmail.com. And I'm happy to chat, answer questions, take suggestions, requests, comments. Catch y'all next time.